Hello, my name is Esther, and I'm a postbac within the NCATS 3D tissue bioprinting group. And let's talk about how exactly new cancer drugs are discovered. So here at the top, I show the drug discovery pipeline, which is crucial for identifying new chemotherapy medications. However, one of the primary issues with this pipeline is that 90% of candidates that reach clinical testing are eventually eliminated. And much of this is due to the steps that are taken before clinical trials, where drugs are tested on two-dimensional cell culture instead of three-dimensional cell culture. And so as such, it is crucial for us to develop more predictive preclinical disease models that are able to mimic the in vivo environment of the human body so that we can better identify drugs with high efficacy. And this ultimately leads to our main objective, which is to develop high-throughput vascularized 3D tissue models that greatly mimic complex human microenvironments, which is where our group at NCATS comes in. So one of our specific goals is to achieve both modularity and versatility. So we want to be able to create a standardized platform that can be easily modified and used for more than one type of disease. And to do this, we first identify organ-specific and disease-specific cell lines, culture them until they are fully grown, and then mix them together in a gelatin solution that we can then bioprint onto a high-throughput platform or a 96-well transwell plate. We then culture this tissue for 7 full days to allow for blood vessel or vasculature formation. After this, we can then add a mesothelial or epithelial cell layer to replicate the barrier function that we see in vivo tissue. And then 7 days after this, we can then add tumor cells to our tissue and track tumor aggregate formation, as well as tumor migration patterns within our vascularized tissue. And lastly, then we can start drug screening to identify drug compounds that are most effective at reducing tumor size in our model. And so on the bottom left here, I show the top-down structure of our platform with each of the aforementioned components labeled. And on the right, I show the confocal image of our vascularized tissue bed, which is in green. And we see successful tumor formation on our vascularized tissue bed, which is in yellow. And here's a zoomed in version of that. So this ultimately validates both our platform and process for use in drug screening. So overall with this platform, we can identify a target, then create disease-specific 3D vascularized tissue, model the disease state, then conduct drug screening all in one platform. And future steps for this work will be to incorporate more immune components as well as fat cells to increase the complexity and accuracy of our model. So ultimately, this project and our preliminary results show that we have the potential to fundamentally improve the way that we approach preclinical drug testing. Thank you so much for listening.